epithelial tissue and bone tissue. The dermis is made of what? What kind of connective tissue is found in the dermis? Both are correct. Loose and dense. Where is loose? Where is dense? Loose is immediately next to the epithelium. You have a thin area of loose connective tissue. And then the most of the space of the dermis, or most of the area of the dermis, is made of dense, irregular connective tissue. It's a very good example of dense, irregular connective tissue, where you find a lot of these thick bundles of collagen. Okay? Of course, there are also reticular fibers and elastic fibers, but to see them, you need special stain. Okay? And the skin is covered by what? The epidermis of the skin. So these are the two layers of skin, dermis and epidermis. The epidermis of the skin contain what? What kind of tissue? Stratified, I'll put the words together. Stratified, squamous, keratinized epithelium. Excellent. And what else is found in the skin?
Hypodermis? Keratin?
Yes, hypodermis is not part of the epidermis. I think there is some dispute about it. Some books they say it is part, some books they say it's not. But usually the two layers of the skin are the dermis and the epidermis on the surface. But what else is found in the dermis that you can find in the dermis? We have seen this. You may see some adipocytes. The hypodermis is mostly made of adipose tissue. The hypodermis, underneath the dermis. It is a fashion that when you dissect a cadaver, you remove the skin, and underneath the skin, you find a lot of fat, yellow fat. This is the subcutaneous tissue. So what else is found in the skin? Glands. What kind of glands? Sweat glands and sebum secreting gland, or sebaceous glands. Okay? And the sebaceous glands are associated with the hair follicle. And the sweat glands are long tubular glands, and they have a long duct which is lined by stratified cuboidal epithelium, two layers of cuboidal cells. And the secretory unit is deep in the dermis or even in the hypodermis, surrounded by fat. Okay? The lungs. We have seen many things in the lungs. What did we see in the lungs? What do you remember about the lungs? Can you list the airways or the air passages in the lung, starting from the big one to the smallest one? Trachea in the lung? In the lung. So you be careful. What is the largest air space you find in the lung? Bronchus, and then bronchiole. And bronchiole, there are different, of course, the bronchi, there are different levels. You may find big bronchus, smaller bronchus. But what differentiates bronchus from bronchiole? Hyaline cartilage. The presence or absence of hyaline cartilage. So if you see a small piece of cartilage, still it's a bronchus. No cartilage, it's a bronchiole. Okay? And then bronchiole lead to what? Lead to alveoli. But before alveoli, you have the alveolar duct. So you have respiratory bronchiole that becomes alveolar duct, and then you have alveoli. And the wall of alveoli is lined by two types of cells. It is simple squamous, but two types of cells. No, alveoli. Pneumocytes type 1 and pneumocyte type 2. Pneumocyte type 2 secretes the surfactant, and pneumocyte type 1, gas exchange. It is involved in gas exchange. Bronchioles are lined by what? Clara cells and ciliated cells. Clara cells and ciliated cells. And it is simple epithelium in the bronchiole. But in the bronchus, what type of epithelium is found in the bronchus? Pseudostratified, like the trachea. It is pseudostratified, columnar, ciliated with goblet cells. Okay? So Clara cells are found in the bronchioles. Now the kidneys. What did we find in the kidneys? What do you know about the kidneys so far? It has simple squamous. Where do you find the simple squamous? In the Bowman's capsule, which is the first part of the nephron. Bowman's capsule is the first part of the nephron. Bowman's capsule surround what is inside? Blood vessels or capillary network. A network of capillaries. And this is the glomerulus. They call it glomerulus. Okay? And the whole thing is called renal corpuscle. And then from the Bowman's capsule, you get proximal tubule, proximal convoluted. So they are convoluted, irregular, which is lined by what?
Simple cuboidal.
Simple cuboidal with or without brush border? With brush border. And this leads to loop of Henle, which is lined by. No, there is no cilia in the kidney. It is lined by simple squamous. The thin loop of Henle is lined by simple squamous. And then you have the distal tubules, which are lined by also simple cuboidal, but no brush border. Okay? Now, so the other organs we will cover after the midterm. So you will see examples of the liver. We will talk more in details about the liver. We will talk about the mammary glands and the uterus as well. Now, this is a summary slide that I showed you before in maybe the second or the third lecture about the processing of a piece of tissue for microscopic examination. So we said fixation is the first step. Never forget this. Fixation is the very crucial step, and it is the first step. And it is followed by dehydration. And then it's followed by clearing and embedding in paraffin. Okay, infiltration and the embedding in paraffin. Excellent. And then you do the sectioning with microtome, and then you have to hydrate the tissue again to do the staining. Hydrate in descending grades of alcohol to do the staining. In H&E, which is the usual stain. And then you dehydrate again to mount. After you stain with H&E, you mount the tissue, but before mounting you have to dehydrate and incubate with xylene because it mixes very well with the mounting medium. And then you mount your slide. Okay? So these are simply the steps of processing tissues for microscopic examination. Now, which step is this that you are looking at here? Embedding. So we are looking at a mold here. This is a mold. This container here, metal container, which has the tissue in the center, the sample, and then it is surrounded by paraffin. So this is embedding. And I'm sure you know which step is going on here. Sectioning. This is a microtome, and this is the serial sections. See, they come like a ribbon. Serial section. And this slide, never forget this slide here. It's very important to remember the mitochondria, the Raphi-R, the Golgi, the lysosomes. So where is the Golgi here? Up and left. So this is a Golgi. And where are the lysosomes? Where do they come from, lysosomes? They come from the Golgi apparatus. And they are found usually around the Golgi or next to the Golgi. But they can be found anywhere around the cell. Lysosome. Now, this is a very nice diagram. It has nothing to do with histology, but it has something to do with the interpretation in histology. So, this is an egg here, which is cut serial sections, and you can see the serial sections here. So, how this egg was cut? Longitudinally. Do you see the yellow part of the egg in all the sections? In all the sections? No. They are all sections of the egg. So this is section of the egg, this is section of the egg, this is also section of the egg. But some of them contain the yellow part or contain the nucleus, and some do not contain the nucleus. So when you look at the section, tissue section, you may not see the nucleus in all the cells. Okay? Some cells may have no nucleus, don't say, oh, this is a nucleated cell. It is like, appears like this because the section did not go through the middle of the cell. Also, imagine that this is a face of somebody, or the head of somebody, head and neck of somebody, and it is sectioned like this. What kind of section is this? Gross anatomy now. It's, how would you, mid-sagittal. It's in the middle here. If it is deviated to the right or left.
Parasagittal.
No, this is sagittal, this is parasagittal. You can say sagittal or mid-sagittal. These are parasagittal. So this is a sagittal section, and it shows you different compartments, the cranial cavity with the brain, the nasal cavity, and the tongue, and the oral cavity, and so on. But if you have if you have a section like this, and this is a slide that you saw in the lab, you remember? You saw in the lab an area like this. The maxillary area here cut, this is coronal section. So we saw this in the lab, coronal section through the head, and we saw the nasal septum in the middle, we saw the nasal cavity, we saw the sinuses, we saw the cartilage and the bone that forms the nasal septum of the nasal structures, okay? So it's important to make sure about the plane of section when you examine a slide. Now here you have a section that shows 2 cells or 1 cell. It shows many cells actually, but it is pointing to 1 cell, 2 different parts of a cell. 1 and 2. 1 is what and 2 is what? It is stained with H and E. Which one of them is stained with H, with hematoxylin? Which one is stained with eosinophil? The nucleus. Why it is stained with hematoxylin? Because it contains acidic structures, and hematoxylin is basic dye. You should not forget this. So basophilia and the acidophilia, and basophilic and the acidophilic. Okay. Now these are 2 sections obtained from the liver. So this is liver tissue, very high magnification. You see only a few cells, and this is also the liver cell, one of these liver cells, showing what? What is number 2 here? This whole thing is what? Cytoplasm. What do you think? This, what is this here? This is nucleolus. And this is the nucleus. The whole spherical structure here is the nucleus. And this is the cytoplasm that contains some elements of rough ER and some mitochondria. These are mitochondria here, many mitochondria. Okay? So, which microscopes were used to take these pictures? Light and electron. So this is light microscopic image, this is electron microscopic image, or electron micrograph and light micrograph. And what is the difference between 1 and 2 here? 1 is pointing to what? Okay, you can say nuclear membrane. If I tell you no, it's not the nuclear membrane. It is slightly underneath the nuclear membrane. It's inside or inner to the nuclear membrane. It is pointing to these structures. What are these? Heterochromatin. Heterochromatin. It's a chromatin, yes, but you should know the difference between heterochromatin and euchromatin. So euchromatin is the pale area, which is here. This is pale area here is the euchromatin. This is the area which has the active chromatin. Okay? And euchromatin is the inactive form of chromatin.